I request everyone to be seated in your places. The technical session 3 will be started shortly.
Have you all got this?
You don't have to be great to start, but you have to start to be great. Now, we will start the session 3 of the day. Now, I would like to invite Dr. T. William Raja sir, Assistant Professor, Department of Biotechnology, to introduce our speaker, Dr. R.S. Suresh Kumar, Assistant Professor, Forensic Technology, College of Natural Sciences, at Arbamanch University, Ethiopia, to the gathering. Please, sir.
Good afternoon, dear participants. I am extremely happy to introduce our guest speaker, an experienced board-certified toxicologist with over 6 years of teaching experience and an able researcher from Ethiopia, Dr. R.S. Suresh Kumar. At present, Dr. R.S. Suresh Kumar is a senior grade assistant professor in toxicology in the College of Natural and Computational Sciences, Arbamanch University, Ethiopia. He earned his UG degree in BHC Biotechnology in the year 2008 from BHC and his PG Genetic Engineering degree in the year 2010 from Bharatiyar University. He obtained his doctorate from BHC for his research work on nano-pesticide formulation, characterization, and thermodynamics in rice model ecosystem. Dr. Suresh is also an American board certified toxicologist with a diplomate of the American Board of Toxicology and an European registered toxicologist. He has done extensive research in toxicology and completed many research works successfully. His expertise lies in human and environmental risk assessment for nanomaterials and microplastics, drugs of abuse and environmental contaminants, detection of pesticides, QSAR models, and forensic toxicology. Currently, he serves as a committee member and East African coordinator at the Society of Environmental Toxicology and Chemistry, American Council. He is also a proud and active member in many toxicology-related scientific communities like American Board Toxicologist, European Register of Toxicology, and International Network of Microbial Ecotoxicology. Dr. Suresh is recognized for his successful supervision of over 18 UG and 6 PG pieces and for holding authorship in 30 impactful paper publications in reputed journals with 850 citations, 13 H index and 14 I-10 index. He has actively contributed to the development of many academic and research setups programs in Arbamanch University. Beyond his academic and research achievements, he collaborates with esteemed professional toxicology organizations, including SOT, STP, SET, TAAFT, holding responsibilities in various capacities. We are indeed delighted to have you with us through online mode, sir. Welcome, sir.
Thank you, Mr. William, for the nice introduction. I hope my audible is. I hope I am audible, right?
Yes, sir, you are audible.
Yeah, I hope. Okay, thank you, sir, thank you. So, good afternoon, everyone. It's my pleasure to be a part of this international symposium, Biotech Spark, hosted by Bishop Heber College, Department of Biotechnology in Bishop Heber College. And many thanks to the organizers for giving me this opportunity. I'm very happy that this is my second time I'm giving a presentation. The first time was, I think, 2 years back. I was in person, I was present in person, and I gave a talk, something related to this, but something little different from this actually. And this is the second time I'm giving the talk to the same department as well as college, so I'm very happy for this, very happy and thankful for this opportunity. So today I would like to present on a topic, it's new approaches and alternatives for toxicity testing in animals. So why this is very important in the sense, now in recent times we see a lot of drugs, lot of vaccines, lot of biological agents are coming into the market, right? So before the agents are coming into the market, it has to undergo some sort of testing, which we call it as preclinical testing. So in preclinical testing, up to I think, to my knowledge, I think up to 2020, up to 2020. We are using a lot of animals for testing. We are using a lot of animals in this preclinical testing studies. But after Corona, exactly after the Corona period, we could see that a lot of people have moved towards a lot of shift has happened mainly in using non-animal. People have started realizing that we are giving a lot of suffering to the animal, and why don't we choose some alternatives for animal? And that's how a lot of changes have come into the pharmaceutical market. And so today I'm going to present you some of the new approaches and alternatives for toxicity testing. Instead of animals, what are the other things we can use in research? This can also be used in your regular undergraduate or postgraduate research. We can use it for any type of research. In any scale of research, we can use this. So, next slide, please. Yes, so what is toxicity testing? Before going into toxicity testing, let us know what is meant by a poison or a toxin. So toxin or poison is almost sounds same. So it is just any chemical that is harmful to the human body. Anything that is chemical or any agent or something that is harmful to human body. It can be a biological organism also, can be harmful to human body. So in general terms, we tell poison as any chemical or physical agent that is harmful to our body, and the study of this is called as toxicology, right? So whatever the person you see in this picture, his name is Paracelsus. So he is considered as a father of modern toxicology. So what is modern toxicology? Before Paracelsus in 16th century, there was some other beliefs regarding poison or toxins or something, but after Paracelsus, after the Paracelsus era, people started realizing that the dose is the one that makes a poison. So what is meant by dose? A dose is a chemical that we give it to the body. So how much chemical we give to a biological organism or how much chemical enter into our body, that will determine whether it is poison or it is a good thing. So whatever the drug we take in our regular day-to-day life, for example, we take paracetamol for fever, we take some antibiotics for control of some microbial infections. So anything we take in the desired amount will cause the desired therapeutic effect. Therapeutic means the treatment effect. Whereas anything if we take in excess amount, above the one that is prescribed or above that is recommended, if we take, it may cause adverse effects. So adverse effects are different types are there. I don't want to go more into what are the different types of adverse effect. Many of you might have studied in immunology also. So anything that we take in excess amount may cause adverse effect, and studying about that is main principle of main objective of toxicology. Right. Next slide, please. Can we go to next slide, please? I think someone has raised a hand. Dr. Merlin has raised a hand. Is there anything important, or should I continue? Okay. Thank you. Fine. So, coming from the next slide. So, what is very important in toxicology is the balance. It's not only about toxicology. Any chemical or any food, water or whatever we consume, everything needs a balance, right? So, if balance is lost, you see what happens to the animal which is there in the picture, right? So, balance is everything, and we should take required amount only for what type of application we wanted to use. If we take in excess, definitely it will cause imbalance in the body. Yeah, next slide. Yeah. So, what does exactly toxicology mean? So, is toxicology a very specialized field, or is it like, is it a specific subject which only some people can study? It's not like that. Toxicology is a multidisciplinary field, that we have to be very clear. It does not include only pharmaceutical industries, because most of the time toxicology is mainly concerned about drugs and other things, but it's not like that actually. Toxicology includes many fields. Yeah, it is a diverse field. Many people from diverse field will contribute for this science. Yeah. Can you next slide, please? Yeah, now we can understand who are all the people who is behind the toxicology. So we have biochemists, chemists, pathologists, veterinarians, engineers, mathematicians, statisticians, animal care providers, animal welfare providers, so even biotechnologists. Of course, whenever we develop a product by biotechnological application, biotechnological means, the product has to reach to the consumer, right? So when it reaches to the consumer may be a human or it may be some other thing. But before it goes to the consumer, it has to be tested for safety. So what is biosafety? Biosafety is simple, that the chemical or the product, whatever you develop, should not cause safety. So most of the toxicologists are mainly concerned about the biosafety of the chemical. So how do they know the biosafety? They try to find out the risk. You know what is meant by risk? The probability of adverse effect, whether it happens or not happen. That we call it as risk, right? So most of the toxicologists will try to find out what is the possible risk which is involved with the chemical or a product or anything we are going to study. That is what the toxicologists will do, and these are all the people who are behind the team. Without all these people, it's not possible for any product or anything to be coming to the market or to develop some guidelines for a product. With these people are very important. So please understand that toxicologist is not only, it does not belong only to the pharmaceutical industry. Every biologist can be a toxicologist if he works at a particular specific application related to toxicology. Yeah. Next slide, please. So what all the toxicologists will do, as I told you, they develop safer chemical products. They try to find out whether the chemical, whatever we develop, is good for the human or good for the environment. They develop risk related to chemical exposure. They also ensure that the food, the water, the basic essential things, whatever we need for human survival, is completely free from any toxic substances. And also, toxicology also has very important application in forensic science. I think many of you know what is forensic science. It's a legal practice. It's a science that deals with, it supports the legal practice. So in forensic science also, toxicologists play a major role. Suppose if any person has taken a poison and died, what poison he has taken. If you want to identify it, we have to go for toxicologists who will help to tell that, who helps to identify what poison the person has taken and he was killed or he died by himself. So for all these things, these are all some of the applications which are done by toxicologists. But there are many other, but I just specified only few. Toxicologists also work in environment. They are called as environmental toxicologists. Toxicologists working related to food industries, food toxicologists. Toxicologists working in regulatory agencies, governmental agencies. For example, Food and Drug Authority, European Public Health Authority. So many authorities where toxicologists will work, who try to set up the regulations guidelines. So this much amount of heavy metal only should be present in water. If it is above 1 gram or 1 milligram, then it is toxic to the human. The water is not good for the consumption. So like that, all these things are designed, or all these things are developed by regulatory toxicologists who are working in the regulatory agencies. So, as I told you, toxicologists is not only do only one specific job, they are multidisciplinary, they work in multidisciplinary aspects. Next slide, please. Next slide, please. Yeah. So now, as I told you before, what are all the different steps in toxicology is explained you. I mean, what are all the different applications of toxicology explained. So in the previous slide, in this slide, what we do actually is, suppose we develop a product, we need to identify two things for a product. One is pharmacokinetics, another one is pharmacodynamics. So if we are using any product related to pharmacy or any pharmaceutical company, we call it as pharmacokinetics. Mostly it is related to drugs, right? So what is pharmacokinetics deals with? Pharmacokinetics deals with the absorption, how the chemical is absorbed by the body, distribution. After absorbing into the body, how the chemical is distributed to different part of the body. That's how, when you take aspirin or when you take paracetamol, your headache is going, because the drug is getting absorbed, distributed in the blood, and it goes to the place where the inflammation is there or pain is there, so it goes and resolves the pain. So distribution is the second aspect, that drug has to be distributed from blood to other parts of the body. Third one is metabolism. The drug or the chemical cannot be present in the body for a long period of time. It has to be removed from the body. That's how the body is designed. So liver is a major organ which does the metabolism, right? So once the metabolism is completed, the drug has to be eliminated from the body. It can be eliminated through different ways. One is through urine, that is by kidneys. The other one is through fecal route. Through gastrointestinal fecal excretion, it will be removed. And also sometimes through sweat, or sometimes through be excreted out of the body. So studying about all this absorption, distribution, metabolism, excretion with respect to time and the concentration, we call it as pharmacokinetics. What is pharmacodynamics? It's a simple thing where once the drug enters into the particular target, how the drug interacts with the target. The target may be a cell, a target may be a DNA, target may be a protein. So once the drug binds to the protein, it causes some effect. That effect is only making you, that is the effect we get once a drug interacts or the chemical interacts with the target. The effect may be two types. One is therapeutic. Therapeutic means the good effect. Suppose you want to treat a cancer, the drug goes and binds to some particular protein and inhibits the growth of cancer cell. That is a therapeutic effect. What is the other one is toxic effect or adverse effect. What will be the adverse effect? Suppose I take the normal recommended dose is 3 gram of paracetamol per day for adult. For 60 kilogram adult, 3 gram of paracetamol per day. Suppose I am taking 10 grams of paracetamol. What will happen? Above 3 grams, 7 grams is excess in our body, right? The bioavailable concentration is very high. So that 7 grams will cause adverse effect in the body, right? So what is pharmacokinetics? What the body does to the drug. How the body reacts to the drug, that is pharmacokinetics. Pharmacodynamics is how the drug interacts with the body, how the drug reacts with the body, that is called as pharmacodynamics. So to study all these things, what is very important is animals. Because directly we cannot test in humans, we have to rely on animals for drug testing. That is very important. Because certain animals which share some of the properties similar to the human thing in terms of evolutionary aspects or in terms of genetic aspects, some laboratory animals share similarity with human being, right? So that's why we use animals in research. And if we see in animals, 34% of the animals are used in biomedical research, 45% of the animals are used in medical and veterinary R&D, research and development research. That's very thing. So you can see major proportion of the animals are used only for medical and veterinary drug development research and drug development or research and development. And for toxicological studies, we use 10%, and remaining is another 10% for some other application. So how do we do the toxicity testing? How do we analyze the toxicity? So whatever the product we have taken, we should take the product. It can be a biological product or a chemical product. For a chemical product, for example, it may be a pesticide. Biological product, it may be a vaccine, right? So what we have to do? First, we have to identify what is present in the product. That's very important. Blindly, we cannot go for a toxicity study without knowing what is present in the product, right? So if it is a chemical product, we should identify what chemicals, what functional group is present. We have to characterize the chemical thing. If it is biological, we should know what protein is present, what is the function of the protein. That things we have to study. So once you know the chemical nature of the product, then you have to go for literature review. Literature review means just search the product in the internet and you can find out in the databases, you can find out what is the nature of the product. Then based on the nature of the product, we can go for structure activity assessment. What is structure activity assessment? Just you know the chemical structure or you know the chemical present in the product, you can just, there are some tools called as chemometrics. Chemometrics or chemoinformatics or quantitative structure activity relationship. There are some tools are available, in silico tools are available, by which without doing the experiment on animals, directly we can find out using the chemical structure what is the probable toxicity of the chemical. We can identify by structure activity assessment method, right? So once you know that, okay, this chemical may cause toxicity to the liver, this chemical may cause toxicity to the kidney, or this chemical may cause toxicity to the skin. Probable, it's not confirmation, it's just a probable identification, qualitative identification, we do it by in silico tool. Once you are sure, then we have to identify how much amount of chemical will cause toxicity. So how to do that? For that, we have to take some animal, which is according, there are some guidelines are there, we have to follow some guidelines and we have to choose the animal, and to the animal we have to give the, in what way we have to give the chemical, we have to give in fixed doses. What is meant by dose? The amount of chemical. So we have to give it in fixed doses for a particular period of time, we have to give to the animal and we have to see what is the response happening in the animal, right? So if you give it for a short period, we call it as acute toxicity studies. If you give it for a long period, then we call it as chronic exposure or chronic toxicity studies. So in this acute and chronic toxicity, we have different types of tests are there, which the best way to remember, best way to explain this is, without toxicity testing, it is not possible for a chemical to reach the consumer or into the product. The toxicity can be for human, or it can be for animal, or it can be for environment. But toxicity testing is compulsory before any consumer product reaches the market. For that, we have to go through these procedures. Next slide, please. Yeah, so as I told you, there are many toxicity testing procedures are there. So we have acute toxicity, we have chronic, subchronic toxicity, we have chronic toxicity. So subchronic means less than 90 days. Chronic means if you are exposing the chemical for more than 90 days, we call it as chronic exposure, right? So what are all the different tests we can do by using this different types of toxicity studies? One is mutagenicity. Mutagenicity is to want to identify whether the chemical is able to cause mutation, it can cause mutation in the DNA or not. That is mutagenicity testing. Then is carcinogenicity testing. You wanted to know whether the chemical is able to cause cancer or not. Or reproduction toxicity testing. There are many drugs, which develop first, but when they are giving it for more than a year or something, it is able to cause some damage to the reproductive organs or the cells in the reproductive system. In that case, the chemical cannot be released into the market, right? So reproductive testing is very important. We call it as DART test, DART testing, which is reproductive testing. Next, we go for developmental toxicity, which is also called DART. Then we go for genetic toxicity. You want to identify whether it's able to cause some change. So we have to do all these things before we identify whether this chemical is good for human use or animal use or environmental applications. So these testings are very important. Next slide. Next slide, please. Yeah. So as I told you, where we can do these studies, as I told you, mutagenicity study. Then there are many tests available in mutagenicity testing. So we can go for simple bacteria, Salmonella typhimurium test, which is called as Ames test, which is a very simple test. Even now it is applicable in many regulatory approvals. They consider Ames test. Then we have a mouse lymphoma assay. Then we have micronucleus test, which is done on human polymorph nuclear cells. Then we can also identify the chromosomal aberration, any changes that happen to the chromosome, whether the chemical is causing any changes in the chromosomal structure or number or anything, we can identify by this test. So in which we can do all these tests. Previously we were doing only on animals. So we, for example, you see ocular irritation testing. If you want to know whether the cosmetic product, I think we use many cosmetic products, perfumes, creams on the skin. How do we know that the cream is not causing damage to the skin? How do we know that? Before it comes to the human market, it is first tested on animals. For example, they test on chicken eye. They take a chicken and they apply on the eye, and they see whether the chemical is able to cause irritation in the eye or not. This you can see very well how much irritation it will cause to the animal when you are doing the testing. The same way, if you want to know whether it's damaging this, if it's irritant to the skin or not, we just shave a portion of the skin from the animal, hair from the animal, like mice, and we apply the chemical, and we'll see whether it's causing any immunological changes or not, irritation or not, we'll see. Some inhalation studies. Suppose you are using a asthma drug, whether it's causing damage to the lungs or not. How do we know it? Before directly testing human, we check it on beagle dogs. These dogs is called as beagle dogs. They are widely used for testing for inhalation studies. So like that, we mostly rely only on animals for doing these studies until 2020 or 2021, until. But after that, can you go to next slide? Next slide, please. Yes. So as I told you, until 2021 we are doing it. But after the before slides, the one before. Please go to the slide one before. Yeah, exactly. Thanks. So as I told you, after 2021, lot of awareness has come among people, and we have seen in many news also that we should stop using animals for research like that. Even India also has proposed some long-term, some period by which they will replace animal like that. They're telling in news, but it's not practically possible. But on what basis people will consider to replace animals in research is 3R principle. 3R principle. So what is the 3R stands for? One is replacement, another one is reduction, and another one is refinement. So what is meant by replacement? Replacement means instead of using animal, why don't we use a system that looks similar to animal? For example, we can use some invertebrate insects in that case, or we can use 3D models of cells. For example, one-dimensional is cell, two-dimensional is tissue, three-dimensional is a kind of organ type. You grow organ in a specific medium in a lab, and you can test on it, right? So we have to find out some alternative methods by which we try to we don't use animals for. That's called as replacement. What is reduction? Reduction is a very important thing. There are some studies where we have to use animal compulsorily. We don't have any choice. In that case, what we can do? Previously, like what we used to do is, we take 5 animals in one group. Like that, we may use 10 different groups and we study. So 50 animals will be tested for a particular chemical or particular study. So what the people 3R group proposes, why don't you reduce the number of animals? Instead of using 5 animals, you try to test it on 1 animal or 2 animals. Right? So there are some guidelines are available by which we can reduce the number of animal we use it for studies. That comes under the reduction principle. We can use some statistical tools. We can use some pharmacological, we can use some pharmacological basis by which only this much amount of, if you know that yes, this chemical is causing this effect means, why you are studying on 4 animals? Just 1 animal is enough to provide the information, right? So that is reduction. And the third one is refinement, where when you are using animal for toxicity research, you should take all effort to prevent the animal from suffering. So that is called as refinement. So you should reduce the pain, you should reduce the stress that you give to the animal should be feeling comfortable while the study is done, right? So all these things comes under 3R guiding principle, which is what now people are considering it very serious, and lot of regulatory agencies like Food and Drug Authority, we have REACH, REACH, REACH Authority in Europe, all this European Union, all are considering this 3Rs as a very important thing in toxicity research. There are some countries which have even told that they won't use animal in research here afterwards. Like that, some countries have also proposed a bill in their parliament, right? So 3Rs are the future of in next 10 years, 3R will be going to be the future in toxicity research. Next slide, please. Yeah, so these are all the international agencies. As I told you, we have first it was started on 2010. Then after that, an international agency called ICATM was developed. This ICATM was one which harmonizes all the alternative methods, alternative 3R based method, whatever developed in different countries. They harmonize all the method and they publish as a the regulator, or they publish as a reference or as a regulatory body. They serve as a central body regulating all this 3R related research. Next is USGPA. They told USGPA, one of the very big agency. They told in 2035 they will stop using mammals in the animal studies. In Dutch, there is a center called as National Committee for Protection of Animal. They told that by 2024 in Dutch, we will stop using animals for chemical research, for testing of chemicals, right? So even some cosmetic product companies, if you see some cosmetic product brand, they just mention that this product is not tested on any animal. Like that nowadays they started to use in the product brand. That also one of the marketing strategy, but it's in European countries, this has become very popular, that if you don't test a cosmetic product on any animal, people are preferring to use that kind of thing. In one way it is good, in other way it's a marketing strategy. Even in India, we see a lot of alternatives has been proposed by the current government. Next slide. Yeah. So how replacement is possible? So as I told you, replacement of animal in US, the same thing you may be using it in India also. The same doctor may prescribe you some medicine and the same drug may be available in India. So in that case, every country cannot have their own regulation or own testing method. So what is very important in this case? Everywhere, every country should have a harmonized approach for doing this replacement thing. What is replacement? The replacing the animals for toxicity research, right? So it takes a lot of time, but effort has already been started. So it is a long-term process, maybe a 10-year or 20 years process, but after 10 years, I'm very sure mammals may not be used in animal testing. Some alternative testings may be proposed, right? Next slide. So what are the alternative organisms we can use? Suppose I am telling that we have many people have proposed 3R concepts. So what are all the things we can use? That's what I am going to explain from the coming slides. So what are all the alternative model organisms we can use? So what are all the spineless solution? What is spineless solution? Invertebrates. Simple way, it's invertebrate. So what are all the invertebrate organisms we can use for 3R principles? Let us see one after the other. Next slide. Yeah, so this one is brain shrimp. I think many of you might have known it, or many of you might have seen in literatures. So what is brain shrimp? It's just a small crustacean. It's a backwater crustacean. It's a brackish water crustacean. It is a small kind of invertebrate, which mostly people use this for giving as a feed for the new fish or something. The new fish, the developing fish is called as fry. From the egg, the fish will develop into small fries, small fishes, right? So for that we give this artemia as a feed, right? So the life cycle is very simple. We can do it very, the experiments, we can easily get this insect for, this invertebrate for research. It's not challenging also, just we need only salt water for the research, right? Next slide. Next slide, I will give you some idea about it. Yeah, next slide please. Yeah, so now we can see what we can do. We just collect the eggs, we grow the eggs are available in the aquarium shops. So you go, you ask for Artemia salina egg, they will give you. Just put it in salt water for some time, like the setup I mentioned here, and we can, and when we allow the eggs to, we have to allow the eggs to hatch. So within 1 day it will hatch, right? After that we see the Artemia, which is a very small invertebrate. We see it in very small size. After 24 hours we get it, and once we get the Artemia, we can put it in microwave plate and we can add our test chemical, and we see how many Artemia dies per particular time or particular concentration. So dose versus exposure, dose versus time. We can calculate in what period of time, in what time the animal is dying, the insect is dying, and how much dose is causing maximum toxicity. Like that we can identify using this small insect. So what is one of the major challenges? This insect grows only on salt water, which means the water is going to be highly saline in nature. So it's not very appropriate for nanoparticle studies or some drug studies, but just as a preliminary aspect, before going into higher animals, we can just test on this animal and we can have a data, and even in environmental research, testing it on Artemia has been widely accepted. The community accepts because it's a marine organism, marine shrimp. We call it a small shrimp actually. So people are accepting it, but not for pharmaceutical research, but at least for environmental research, we can test on Artemia and get some idea what is the toxicity of the chemical on the Artemia cyst. Next slide. Yeah, so you can see this was from my PhD work where I developed one nanoparticle, nano form of pesticide. So I given this chemical to the Artemia cyst, right? And after some time I just observed the chemical under the I just observed the insect under microscope, dark field microscope, and I was able to see if the chemical was able to cause some damages to the insect to the invertebrate or not. I was able to identify, right? So by this way, one data we can get is how much dose of the chemical is causing how much concentration of a chemical is causing toxicity, we can identify. Second one, what are all the changes that happen to the insect because of the chemical? Just morphological changes we can observe very easily, right? So anyone can do this. It's not very expensive method. Still scientific community accepts this kind of studies. Mainly for environmental research we can use this insect. Next slide. Next is Daphnia magna. So Daphnia magna is even now a well-renowned toxicological model system for ecotoxicity studies. Suppose you want to know the product or the chemical or the nanoparticle or any biotechnological product or anything you developed, whether it is causing problem to the environment or not. So which is a representative organism for the environment? Representative means one organism if you study, that gives you idea that whether it will cause harmful to the other organisms also, right? So to know, because the environment has many organisms, many biotic species are there, how do we know which organism to study? So it's better we can go the widely known, well-established toxicity model called as Daphnia magna. Many of you know it, it is available in the pond. Only thing is we should know how to isolate the Daphnia magna. If you know how to do it, it's very easy. The insect is transparent, first thing. All the inner structures of the insect can be clearly visible, clearly identified, right? And it's very easy to maintain in any lab. You don't need any big setup to do it. So Daphnia magna is even now is considered a well-known model for ecotoxicity studies. Next slide. So, how do we get this insect? We can get it from market by this kit called as Daphnia toxic kit, which is a commercial kit, but commercial kits are always expensive. If you don't want to do a kit, you can just go to the pond like this, like I mentioned there. Then you use a separate filter, membranes, you can use a steel filters with specific size. For example, 50 micron or 100 micron cutoff range you can use, and you can filter the pond water. And the Daphnia is having a fixed size, so it will get retained on the filter and the water will be filtered out. And once you retain the organism on the filter, you can transfer it to a medium. A simple cultivation medium is there. You can transfer it and you can observe under microscope whether it is Daphnia or any other organism. If you are able to identify Daphnia selectively by using a pipette, siphon pipette, we can take that organism and grow it in a separate media. They develop it, and over a period of 3 or 4 days, we get a pure culture of Daphnia magna. Right? So it's easily we can do it. Any chemicals, we can easily study on it. Nanoparticles, excellent model system for studying the toxicity of nanoparticles. Right? So widely accepted by the community, widely accepted by the regulatory bodies as well as in the scientific community. Anyone can do this experiment. Next slide. Yeah, so this is just a paper I gave an example just for to understand how. So this Daphnia magna is used as a, in a food chain we have primary consumer, we have primary producers, consumers, in consumers, primary consumer, secondary consumer, then tertiary, right? So Daphnia magna can act like a primary consumer, right? So the Daphnia magna will act on some bacteria or some particles present in the water. So as a primary consumer, you can use Daphnia magna as one of the system, right? This Daphnia will be feeded by, Daphnia is a feed for secondary consumers like little bigger animals, aquatic animals, for example fish or some small bigger invertebrates. They feed on this Daphnia for the survival, right? So if you take a small tank setup, in that you take your Daphnia, you grow Daphnia, and you grow another organism like a small fish or something which consumes Daphnia. So in a simple setup, we are able to create a mesocosm or a simple simulatory ecosystem based simulatory experiment. We can do it in a small tank setup. Yeah? So that's why it is widely used for many of the ecotoxicological studies. I have given some reference like that, lot of references are there. Next slide. Yeah, next I want to highlight zebrafish. I think many of you might have known what is zebrafish, right? It's an aquarium fish, but nowadays it's not aquarium fish. No more it's aquarium fish. It's used for a lot of medical research, right? So what is the significance of this zebrafishes? The development of zebrafish from egg to the one particular stage is easily observable. Like, which observable means, we know that in cell biology we studied about different cell stages, right? So all the cell stages which is happening can be easily identified in the zebrafish, the zebrafish, developing zebrafish, right? So it is a widely used organism for developmental toxicity studies. Not only developmental toxicity, even all human disease models has been created in zebrafish. For example, atherosclerotic model, myocardial infarction model, neurological Parkinson model. So all disease models which we see in human can also be created in the zebrafish because we share some of the genes similar with the humans, right? So a lot of databases are there related to zebrafish genes, proteins, knockout experiment. Many things have been done in zebrafish. So even now it's widely used to model. Only thing we need is we need a little setup to maintain the fish, but other things are all easily accessible, easily available in internet. Our reference sources are available to do testing on zebrafish. If you do a study on zebrafish and Daphnia, it's more than enough to find out the ecotoxicity of a chemical. Next slide. Next slide. Yeah, so here you can see how zebra